All right, let's get started with the afternoon session. So in this session, we have actually 3 talks. The first one is by Philip Schlegel from University of Cambridge. And he's going to talk to us about connectomics. So the whole session is about computational neuroscience and multi-scale modeling. After Philip, Brian is going to talk to us from BU University. And he's going to talk to us about odors and some cool stuff that he's doing over there, but we'll get into detail of that. And after that, Mariam is here from University of South California, and after her talk, we'll have some break. Then we'll come back here. We're deciding how much we want to have the break, and then we'll come back here for a wrap-up synthesis discussion, and then the panel discussion. All right. So with that. Any further ado, please.
Thank you, and thanks for having me. I've thoroughly enjoyed the symposium so far. So in the lead-up to the symposium, Nima asked me to, wouldn't it be nice if you discussed the, Fly Connectomics ecosystem, the tools and resources, and I was happy to oblige. So the task for my talk today is to go broad and tell you, all the bits and pieces that are out there, and hopefully whet your appetite a little, and maybe by the end of it, some of you might consider doing some modeling on the fly. So I'll start off by telling you why I think the fly is a cool model system for various purposes. And first of all, the fly central nervous system is complex, but not too complex. So we're looking at about 160,000 neurons across the central nervous system, of which about 40,000 are in the central brain, and you've got about 50,000 each of the optic lobes. Those are the purple bubbles to the side, and then about 20,000 neurons in the ventral nerve cord. So this might already look familiar from some of the talks from yesterday. Fly neurons, and that's true for insect neurons in general, are fantastically diverse. Here's just a quick slideshow. This is flywire data, and I'm showing you sort of a random sample of different kind of neurons. Our running tally for the number of cell types in the fly brain are, and we're revising this number as new data comes in, is about 8,000 cell types in the central brain, and about 600 cell types in the optic lobes, right? In the optic lobes, you've got this repeated structure of the visual column, so slightly less diversity there. The important thing is that, like most insect neurons, these are highly stereotyped. So we're able to reliably find the same neuron again and again across individuals, mostly based, or in a lot of cases, based on the morphology alone. Right, so the other piece to that puzzle is that we have genetic access to a lot of these cell types. Case in point, there was this paper just recently from the Flylight project at Genelia, where they describe more than 3,000 genetic driver lines that let us target individual cell types. And that you can combine it with effectors, right? So in the little light confocal stack on the right, they use the driver lines to put GFP in those neurons. It's just 2 of the cell types in that collection, but you can use it to put in calcium indicators or optogenetic tools to actually manipulate neuronal activity. And if you're a wet lab experimentalist, you would go on this website down there to browse the collection, find interesting lines for your purposes, and then order those fly strains from Bloomington. And depending on how far they have to be shipped, in a few weeks you've got the neurons as the flies that you can do your experiments with. Right. So we've got sort of the tools, right, to experimentally probe the fly's nervous system. The question is there interesting behavior? And I sort of semi-randomly picked courtship. So what you see here on the left is a video from Vanessa Ruta's lab, where they're tracking 2 male flies courting the same female flies. The same one female fly, and they sort of go back and forth between courting and fighting each other over the female, and this seemingly simple behavior has been studied extensively for various purposes. So people have studied action selection. That's actually what the video. What the video of Vanessa Ruta's lab is on. They've studied motivational dynamics, visual feedback, motor control, and so on and so forth, right? So sort of a plethora of different neuronal mechanisms you can study. And if fly courtship isn't doing it for you, there was this recent paper, the preprint had been out for a while, but it just got published in Current Biology, and I thought it was so cute that I'd had to at least mention it. So this is a paper from Wolf Hitterroth's lab, where they designed these little arenas for flies that contain both a food source and a little like rotating platform, right, a carousel. And it turns out that some flies, but not all flies, really enjoy being flung around. And that, you know, that you can see that on the right. And they actually prefer that over having food. So this is essentially sort of an idiosyncratic play-like behavior. It just goes to show that the fly can surprise even people that have worked with it for years at this point. So we have interesting behavior and interesting underlying computations that we might want to investigate. And we have the experimental access. So now we need something to sort of tie this together, right? So how can I come up with a circuit hypothesis that I can then experimentally test or model? And this is where the connectome comes in, or connectomes in general. And yesterday, Ariel and I had a discussion over lunch about how the term connectome potentially can mean something slightly different depending on your background and the kind of data you work with. So in the fly field, when we say there's a connectome, what we mean is a comprehensive anatomical inventory of all neurons in the brain and the synaptic connections between them. And what this looks like in practice, so essentially the first step is to acquire a nanometer resolution image data set that currently means electron microscopy, but there are a couple of contenders that are on the horizon. Expansion microscopy, where you then can use light microscopy to acquire the data set, but also X-ray tomography are contenders that might be an alternative solution for that in the future. But no matter how you acquire the data set, the next step is to segment it, segment both the neurons as well as the synapses in the data set. And from there on out, the next step would be at least currently human proofreading, right? So this is one of the major bottlenecks currently when it comes to generating connectomes. The reason is that the initial segmentation isn't perfect, and in order to make it useful for researchers, you have to fix the errors that the machine learning algorithm has done, or the neural network has made. And sort of tied in with that is sort of a bit of a recurrent feedback loop. You have to make the data set indexed, right? So you have to add annotations, salient labels, so that people can actually use it. You don't want to work with just some sort of segmentation ID. Okay, so that's sort of the setup, how you get to your connectome. And obviously there's sort of a multi-year process. Just sort of to give you some ballpark numbers for the hemibrain data set, which was the first sort of large-scale connectomic data set for fly brain. The proofreading alone was 50 human person years, right? Okay, so then looking at sort of a timeline of connectomes in the fly, the first connectome not in flies, but in C. elegans, famously was generated by Sidney Brenner, actually at the LMB in Cambridge. Very proud of that history. And then it took more than 20 years for the first, for the next connectomes to basically pop up. So you can see that in the 2010s, we had a couple of smaller partial volumes connectomes, one of the optic lobe, one of the antennal lobe, that's sort of the olfactory bulb analog in the fly, and one of the mushroom body, that's the the Center for Associative Learning Memory. And then only in 2020, the hemibrain was published, right? So that's a data set generated at Janelia. As you can see from the little schematic, it comprises about half a hemisphere and like a chunk of the optic lobe, of one of the optic lobes. Then in 2021, the lab of Wei Li in Harvard published a fancy data set. It's a female adult nerve cord. I actually coined that. They had a contest where they asked, like, what should we call it? And fancy just sounded like a good acronym. And after that, the groups of Albert Cardona and Marta Sladic, who were originally at Janelia and are now in Cambridge, published the connectome of the larval brain. And then the next in the sequence was the Mancy data set. So they sort of stuck with the fancy Mancy, right? So this is now the male adult nerve cord. There was again a Janelia data set generated in collaboration with us in Cambridge. And then late last year, we then published the 5Y data set that was spearheaded by the labs of Mala Murthy and Sebastian Sang in Princeton in close collaboration with us, and a range of other crowd science project, a range of other groups across the globe. Right, so if you look at that sequence, right, so what's the next logical step, right? So you've got a half brain, you've got a nerve cord, you've got a larval brain, and you've got a full brain. Next logical step is a whole nervous system, like from one animal. And indeed, lo and behold, there are currently 2 data sets where people put the finishing touches on. One is the male central nervous system. That is, again, a Janelia data set that we're working on together with Janelia. That's probably ETA's like early summer, so it's really around the corner. We're starting to write up a paper now. And the second one that I'm less familiar with, I know of it, is the bank data set, brain and nerve cord. That is being generated again by the labs of Wei Li, and Rachel Wilson is involved, and it's supported by the Flywire folks in Princeton. Okay. And just to sort of like, this is the piece de resistance. That's our male nervous system here, again, generated in collaboration with people at Janelia, Stuart Burke, Jerry Rubin, and with a lot of help from Google, who did the segmentation for these data sets. 166,000 neurons, more than 120 million synaptic connections, and that's something you'll be able to play with if you want in just a few weeks. So there's a connectome. What kind of data do you get from it? I guess it's sort of in the name, right? So obviously you get a graph where a node is a neuron, and an edge means a synaptic connection. But actually, if you only work with a graph, you're throwing away a bunch of data, for better or worse. Because in reality, what the connectome actually contains is morphologies for all the neurons, right? So, we're going from voxels to meshes to skeletons, whatever representation you want and you can generate. And on top of that, we've got, basically layered on top of that, we've got the synapse detection, right? So we know for each neuron where the inputs, where the outputs, and then because of that, because we know the pre- and post-synaptic partner for each of those connections, we know for any given connection between 2 neurons where those connections are on the dendrites and the axon, respectively. Or I guess, there's axonic connections and dendritic. So essentially, we've got this sort of really rich data that I think sort of lends itself to the kind of multi-scale modeling that we might want to do. Okay, and some of you have at least heard of that, and I know Jakob mentioned it, at least briefly in passing. Turns out you don't only get these sort of anatomical data from the connectome, or you can get more than that. So back in 2019, there's this recurring conference, Connectomics conference, that is 1 year in Berlin, and the other one is at Janelia. And 2019, it was in Berlin, and after the conference, right, that I've got this last hurrah on the last night, and at the next day, Jan Funke and Alex Bates and I were sitting in the beer garden, and we were sort of slightly hungover and discussing sort of next cool projects. And someone pitched, well, wouldn't it be cool if we could get neurotransmitters from the connectome, right? This is one of the things that you need, right? You want to know, at least, is the sign positive or negative? Is it excitatory, is it inhibitory? So, we were like, sure, let's give it a shot. So Alex went in and essentially collected this data set, this ground truth data set for neurons where we know the neurotransmitter based on past literature, and then identified those neurons in the EM. And then Jan Funke, who's at Janelia, and his then PhD student, Niels Eckstein, they took little cutouts around the individual synapses from the EM image data and trained a neural network on it. And you can see it sort of from the confusion matrix, right? It just works. So to everyone's surprise, I think, or at least to my surprise, it worked really well. And ever since, as far as I know, all the major connectomic data sets in flies have this data attached to it. So you know for each synapse what the predicted neurotransmitter is, accuracy, and so confidence, and then usually it's aggregated per neuron. All right, so we have this beautiful connectomics data, but in able to sort of maximize the use, we have to be able to connect the connectomics data with the driver lines and with other extant data, right? Like there might be a physiological data set around that you might want to include. And people have thought about this kind of problem, so I'm just going to briefly mention 2 tools that are, that people use to connect the dots. One is the this tool called Neuron Bridge, also developed at Janelia. It's essentially an image-based search where you can go in and you can put a neuron ID or a cell type from the connectome in, and it tells you which of the genetic driver lines might contain your neuron, so what might give you access, and you can do the inverse as well. So if you just go to that website, you can have a look. And the second thing is Virtual Flybrain, which is a knowledge database. It's funded by the Wellcome. It has a bunch of curators that are doing this full time. So what they do is they essentially take image data, publications, they index them, they cross-link them. So if you're lucky and there's some research on a neuron that you're interested in, you can just go type in the cell type again, or even the ID from the connectome, and it tells you, look, there's these 3 publications, and this is kind of what we know about the neuron. So this is a really nice way of sort of like, if you don't know a whole lot about the thing that you're interested in, that's a good starting point. All right, so what do people use the connectome for? Our group, at least with the current new data set, the male CNS, is interested in how do brains compare, right? And we just happen to have a male and a female fly brain, so the hook for the up-and-coming paper is essentially looking at differences and similarities between sexes when it comes to synapse resolution, fly connectomes. But given the audience and sort of there's a limit in time, I'm instead going to sort of just point out a couple papers and projects that I think are really cool and that use connectomic data to model brain circuits at various scales and sort of to gain mechanistic insight into or like learn about the dynamic function. So we already heard yesterday, right? So you heard about the model of the visual system from Jakob's lab, and then Bing told us about this embodied model, and the thing that I really like, this is probably my favorite piece from this symposium so far, is how people in John Tuttle's lab were able to take the connectome of the nerve cord specifically and identify a potential CPG for walking. So if you like these papers, I would suggest you have a look at this also very recent. I picked recent things because I thought people might actually appreciate it more than me rehashing papers from whatever, like 4 or 5 years ago. But there's this recent paper from Tom Clendenon's lab where they essentially look at the predictive power of the connectome. So like, what can the connectome actually tell you about? And they sort of run, it's focused on the visual system, and they run it through paces. So essentially try different assays. This is this little panel here is where they tested whether you can predict the receptor field of a neuron based on just the connectome itself. So have a look at that if that sounds interesting. And then the other thing, sort of as a counterpoint to these like large-scale models, right, where it's almost the whole brain, or at least sort of a large brain region, there's another paper that also came out just a few weeks ago. It's a preprint that came out a couple of weeks ago from Sebastian, well, not Sebastian's group, from Sebastian himself, where he models individual neurons, again in the visual system, but essentially what he's looking at is whether it's a good idea to approximate even small insect neurons as point processes. And the gist is, without giving too much away, have a look at it, is yes and no. So some neurons is fair enough to approximate as a point process, but other neurons, generally not a great idea, or probably not a great idea. Okay, and I'm going to finish with probably the most important slides, which is, if you're interested, where do you get the data? Where you get the data from depends a bit on the data set. So all the Janelia data sets, which currently include the hemibrain, the male nerve cord, and the male CNS when it comes out in a few weeks, you can get from Newprint. All right, if you just go to newprint.janelia.org, you can log in with a Google account. You can interactively browse those data, and there's Python and R tools to download it programmatically if that's what you want. And then for Flywire and the up-and-coming bank, where I'm not quite sure, I'm guessing also ETA sometime in summer, you would go to codex.flywire.ai. Again, log in with a Google account and you can download the data and explore it. And for the larval brain, that's hosted by the Virtual Flybrain, so that if you go to catmate.virtualflybrain.org, there's a number of data sets, and you just sort of have to find the L1 EM larva, I think is what it's called. And last but not least, if you're interested in any of this, I suggest you have a look at this little summary of the tools that we've developed in the group. right? So we have a range of different R and Python tools that essentially help facilitate working with this data, both to just get it, but also to process it and analyze it. And with that, I'm at the end. I've got 10 seconds left. Thank you for your attention. And there's a whole bunch of people to thank, both in Cambridge as well as in Princeton, at Janelia, of course, and even at the Allen, where Forrest and Casey and Ben and Sven are actively working towards not just maintaining the whole IT infrastructure that we use for these connectomes, but also develop it. Thank you.
All right, questions? All right, I have a question myself. So, you mentioned the episode where drinking ended up into, tagging, the neurotransmitters. Is there a way of also, going into the subcellular space and figuring out, for example, what sort of, organelles are in this sort of, neurons, because this is much smaller than, let's say, mouse brain, right? Let's say you can figure out, okay, there's ATP-related sort of systems like mitochondria, blah, blah, blah.
There is for the hemibrain at least, I'm not sure whether they're planning to do this for, say, the male CNS, but there is an organelle segmentation. I didn't mention that, but yeah, you can at least for the hemibrain, look at these things.
Okay.
And there's also, they tend to have nucleus segmentations if you're interested in that kind of thing. So yeah, that you can do that, and for some it already exists.
Okay.
Some of those data sets.
Okay, cool. All right, other questions? Eric?
So just, thank you very kindly. Just with a nod towards your linkage between the connectome and dynamics, we had two spectacular lectures at this symposium which took very simple models of point neurons, either the rate-based models of Brunton and Tuthill and collaborators, or the similar sort of linear voltage equations of Maca. To what extent do you think there is a large distance to travel or a lot to be gained by thinking about really complex and diverse dynamics at the level of individual neurons and synapses in Drosophila, sort of versus other species that we might be more familiar with, mammalian or other species?
Yeah.
Thank you very much.
It's a good question. I don't think I've got a great intuition. The only thing I would say is that I think we'll get a long way, hopefully, with just sort of simple point process, whatever, firing rate models, right? And then eventually you'll hit a wall where, we just can't explain this phenomenon just based on this simple model. And at that point, we may actually have to go in and, look at the, whatever, what happens in dendrites, the network, firing rate models, for example, don't distinguish, at least as far as I know, between exodendritic connections, exoxonic connections, and so on and so forth. So we'll basically have to add detail as needed. But I can't tell you where, at what point we'll have to start doing that. But the data's there, right? That's the encouraging thing.
I'm a little bit. I would have imagined there here.
Oh, yeah.
Many
sorry.
counterexamples, right? The looming response. There's no way you can get that from point neurons, right? Or a lot of the stuff that's happening in the central complex.
Yeah.
And so...
Why not the central complex?
there's a lot of... The action happens in neuropil, quote unquote, right? Who knows what's going on there, but, that's where people record from, and so that's... So, I don't know. It seems paying attention to dendrites and neurites is going to be really, really important.
I agree.
Often it's like inputs and outputs in the neurites, so it's
Yeah.
like non-canonical. It's the rule rather than the exception.
I fully agree. There's a spectrum where insect neurons can be very polarized, where you've got mostly inputs in the dendrites and mostly outputs in the axon. But the reality is that a lot of the neurons are fairly mixed, and surely there's a difference between, as I said, whatever, dendrodendritic connections and exoxonic connections. And as a data point there, we quantified this recently, although, don't quote me on the exact number, but it was about 30% of all connections in the flybrain are exoxonic, which I think just goes to show that, they're important at the very least.
Maybe an unrelated question to how reconstructed are the most reconstructed of these volumes? When people talk about connectome, what does that really mean in terms of, if I take a random synapse, a random pair of neurons and I dig in, what can I expect?
I mean, okay, so this is sort of the dirty little secret of Connectomics, right? Like when people declare victory, that just means that they run out of money to proofread, or it's sort of, you hit sort of diminishing returns. Which is not to say that, like there isn't sort of a rhyme and reason behind it, but essentially you have these sort of free-floating synapses, right? The synapses are essentially detected, and you then have to associate them with a neuron, right? And that's what you do. That's what you do the proofreading for, to fix up the neurons so you can associate the synapse with it. In reality, when you're at the end of the day, you end up with somewhere in the realm of, like, let's say 50%, right? It depends on the data set, it depends on the part of the area of the brain, and the quality of the image data, yada, yada, but 50% of the synapses, we don't know what they belong to, give or take. That's just the reality of it, right? So, but again, on the plus side, we know what you don't know to some extent.
Is there any effort in trying to like systematize the, the sort of morphological structure that we get from connectomic data, or to like some sort of like equivalent graph net? You mentioned the graph, that you said the graph is a simple thing, but is there any effort that you know of that people are trying to?
This is precisely what Sebastian tried and suggested in this preprint, right? Essentially says, like, look, maybe we shouldn't think about synapses, but we should think about sort of, I forgot what you called it, like effective connectivity, right? So you're basically turning the synapse weight and the synapse position and the morphology of a neuron into something that's more biophysical.
Okay.
You can think about it as, this is sort of not quite accurate, right? Like, sort of depolarization at the action initiation zone. Sorry, action potential initiation zone. Okay. But yeah, I think that's where we want to go eventually, right? Improve the graph representation.
All right, let's thank Philip